Hey guys, this video is going to go over the process of cell respiration. I'll talk about glycolysis, the Krebs cycle, and oxidative phosphorylation. So I hope this is helpful. This is the mini poster I'm going to have each of you create for cellular respiration. It has the general overview, plus for each step it has what goes in, what comes out, and then what is net produced. Start by making your title and the general equation for cellular respiration. So this is going to be C6H12O6, which is glucose, plus 6 oxygen molecules is going to produce 6 carbon dioxide molecules, 6 molecules of water, and then energy in the form of ATP. The next part of your poster is going to be the general overview of the process. So on the left-hand side, please glue or tape on the paper that was provided to you. On the right-hand side, write the corresponding number for each process. So glycolysis is step 1, Krebs cycle is step 2, and oxidative phosphorylation is step 3. You want to make sure that you include the small writing that I have listed and the number of ATP. At the bottom, include the total number of ATP produced for each glucose, which is going to be 38 ATP. And on the picture on the left-hand side, make sure you take the time to write all of the small print. There are a couple of things on this picture that I want to point out before we move on. The first is that step 1, glycolysis, it's happening outside of the mitochondria. And then you can see an arrow going from the cytoplasm, which is where glycolysis occurs, into the mitochondria for step 2, which is called the Krebs cycle. So glycolysis is either going to move into cellular respiration for that aerobic process as long as oxygen is available. But if oxygen is not available, then this is going to lead into the anaerobic process called lactic acid fermentation. Underneath the picture, go ahead and add the first step, which is glycolysis. Like I said before, this occurs in the cytoplasm, and it can either make way to the aerobic process, which is called cellular respiration, or it's going to go into the anaerobic process, lactic acid fermentation. I just abbreviated it with an LA on your paper. For the substances that go into glycolysis, first you need 1 glucose molecule, which you wrote above as C6H12O6. You also need 2 ATP to get the reaction started, along with 2 NAD+. NAD+ is going to be your electron carrier. You learned about that yesterday when you looked at redox reactions. That plus sign indicates that the NAD can accept a hydrogen, which is going to be what we refer to as when we talk about having an electron. So it's going to be what donates an electron when we get to that last step of oxidative phosphorylation. The products that are produced from glycolysis are going to be 2 pyruvates. Essentially, you take a glucose and you cut it in half. You'll produce 4 ATP and then 2 NADH, which is your electron carrier that is now charged with 2 electrons. Because glycolysis required some ATP to go into it, the net amount of ATP that is produced is 2. It required 2 to start, you produce 4, which gives you a total of 2 ATP that the cell can now use for energy. Before we move on, I just want to draw your attention back to the picture at the top. So that first step, glycolysis, it shows that glucose is broken into pyruvate. Remember that you make 2 of them. Those are going to enter and power the Krebs cycle. If you look at the top where it says electrons via NADH, those 2 NADH that were produced, they're going to go over into the mitochondria, and they will be used in step 3 when we get to the electron transport chain. Step 2 is called the Krebs cycle. It's also referred to as the citric acid cycle, so make sure you know both names. In this process, we're moving from the cytoplasm into the matrix of the mitochondria. For each pyruvate that enters the Krebs cycle, it's converted into acetyl-CoA, and then it'll go through the process of producing 3 carbon dioxide molecules, 4 NADH electron carriers, 1 FADH2 electron carrier, and 1 ATP. Since 1 glucose molecule produces 2 pyruvates, this cycle is going to happen twice. So what you're actually going to net is 6 carbon dioxides, 8 NADH, 2 FADH2, and 2 ATP. Looking back up at the picture, in step 2 of the Krebs cycle, from the pyruvates that go into that cycle, we produce FADH2 and NADH, which are going to carry electrons to step 3 of the process, where we find the electron transport chain. Also, underneath the Krebs cycle, you'll see that there's an arrow where there's ATP coming out. Again, we produce 2 ATP, and then from the cycle, 6 carbon dioxides are produced as waste that the cell will then get rid of. The third and final step of cellular respiration is oxidative phosphorylation. This step happens in the inner membrane of the mitochondria. There are 2 main parts of oxidative phosphorylation. There's the first part called the electron transport chain. I abbreviated it as ETC. The electron transport chain uses the 2 NADH that came from glycolysis, the 8 NADH and 2 FADH2s that came from the Krebs cycle to pump out hydrogens to create a gradient that's going to power the second step of oxidative phosphorylation. The gradient that's created with the hydrogen ions from the electron transport chain is going to power the second step called chemiosmosis. Chemiosmosis is a process where the hydrogen gradient drives ATP production by powering ATP synthase. ATP synthase is an enzyme that produces ATP. So what comes into oxidative phosphorylation totaled up is 10 NADH, 2 FADH2, and remember that those are the totals that came from step 1, glycolysis, and step 2, the Krebs cycle, and then 6 oxygen molecules that came into the cell. We finally see oxygen playing a role in this process. The electron carriers that come into the electron transport chain are what drive ATP production. For each NADH that comes into the electron transport chain, it's going to produce 3 ATP, and for each FADH2, it's going to produce 2 ATP. So if we multiply those by the total amount that go in, so 3 times the 10 NADH that come in, we get 30 ATP, and then 2 times the 2 FADH2 that come in, we get 4 ATP. Total from the process of oxidative phosphorylation, we'll see 34 ATP that are produced. So the total that comes out of oxidative phosphorylation are 34 ATP and then 6 water molecules, which are just a byproduct of the process. So looking back up to the top of your poster, the overview at the top of step 1, 2, and 3 show you that 2 total ATP come out of glycolysis, 2 total ATP come out of the Krebs cycle, and then from all of the byproducts of the electron carriers that are produced in the first step, we get 34 ATP that are produced in oxidative phosphorylation, which includes the electron transport chain. For 1 glucose molecule that goes through the process of glycolysis, the Krebs cycle, and oxidative phosphorylation, this cell will produce about 38 ATP that can then be used for whatever process is necessary. This poster is meant to be an overview for you so that when you go to study cellular respiration, you can look back at this and clearly see that in each step, you know what goes into it, what comes out of it, what is net produced. You also need to know where these processes are happening. Lastly, you want to be able to trace where the ATP comes from in each process, including the electron carriers that came from glycolysis in the Krebs cycle to power the electron transport chain. Tomorrow we will look more closely at oxidative phosphorylation and how the electron transport chain works, along with how it creates the hydrogen gradient to power chemiosmosis, which is the main production of ATP that the cell has. For now, use this information and the information that you got in your guided reading to help you work through the cell respiration packet that I've left for you. I hope this was helpful. See you all tomorrow.